Hi, ladies and gentlemen. I'm going to walk you through how to do the using models in DNA structure for DNA structure and replication activity. Okay, so let's start off looking at the first slide. It says DNA structure and replication. How is genetic information stored and copied? Okay, so deoxyribonucleic acid, or DNA, is a molecule of heredity. It contains the genetic blueprint for life. For organisms to grow and repair damaged cells, each cell must be capable of accurately copying itself. So how does the structure of DNA allow it to copy itself so accurately? So then we're moving on. So that's our key question. So how is the structure allowing the DNA to be copied or replicated? Okay, so on slide 2, this is our first model that we're using for our lab today. You'll see that the model has 4 parts. There's the nucleotide portion, okay, up here. Okay, then there's the nitrogen bases that are coded for on the bottom left. Then we've got the ladder model of DNA and the helix model of DNA. Okay, so we are going to be referring back to slide 2, so you'll have to flip from your from slides 3, 4, 5, and 6, okay, back to slide 2 to help you answer these questions. Okay, so let's take a look at slide 3. Okay, it says, what are the 3 parts of a nucleotide? What kind of sugar is found in a nucleotide? Which nucleotide component contains nitrogen? And then name the 4 nitrogen bases shown in the model. Sorry. So when we are looking at our model, okay, so the first question asks, what are the 3 parts of a nucleotide? I'm going to use this diagram here. So the 3 parts of the nucleotide, you'll see there's the circular part, there's the pentose or pentagon shape, okay, and then there is kind of what in this example is kind of like a half an oval shape that's attached. Those are the 3 parts to a nucleotide. Hopefully you remember this from last week. And we've got a phosphate, a sugar, and a base. Okay, the second question is, what is the kind of sugar, or what type of sugar is found in DNA? Well, the D in DNA stands for the deoxyribose sugar. And then question number C, which nucleotide component contains nitrogen? Okay, so there are 3 different nucleotide components. Okay, this one is described as the nitrogen-containing base. So the bases are what contain nitrogen. And then what are the 4 bases that are in DNA or in our nucleotides? There are 4 different bases that are possible. We have adenine, thymine, guanine, and cytosine. So those are the 4 different bases that we find in DNA. And you'll see each of those bases is coded for with a different shape. Okay, so you should now be able to answer 1A, those are the 3 parts, phosphate, sugar, and base. Okay, the type of sugar, what kind of sugar is found in the nucleotide, a deoxyribose sugar. Okay, which nucleotide component contains nitrogen? Well, it's a nitrogen-containing base, so all of these bases contain nitrogen. So you should be able to answer 1, A, B, C, and D. Okay, then we're moving on to slide 4. Okay, in slide 4, it asks you to circle a single nucleotide on each side of the ladder. Okay, so I'm going to come to my picture here. I'm going to go to the circle, okay, and I'm going to move the circle so that this is a nucleotide, right? So a nucleotide has those 3 components. I'm just going to move that over to my DNA ladder, and I see that, hey, this is 1 nucleotide, a phosphate, a sugar, and a base. I'm going to make it a little wider to include the actual base here, okay, and a little skinnier, so it's just 1. Okay, and then I also need 1 on this other strand. Okay, so connected on the other strand. Okay, again, I need a phosphate, a sugar, and a base. And in this case, you'll see the phosphate is here. So it's phosphate, sugar, base. This is not the phosphate that's attached to this nucleotide. Okay, so you'll see that the phosphate kind of comes off the upper left-hand corner of the nucleotide. And when I build my DNA model here, Hey, this nucleotide is flipped upside down, so it's pointing down. That means the phosphate is this lower phosphate, not the upper phosphate. So it's super important to recognize, hey, the phosphate that's part of this nucleotide, not the next nucleotide. Then you are saying what parts of the nucleotides make up the rungs of the ladder. Okay? The rungs of the ladder would be like where you would put your feet and your hands. So it's what's making up the middle section, or where you would put your feet, or the steps of the ladder. So what makes that up? If I look at these two shapes here and these two shapes here, I find them over on the left-hand side of my key. What are those that are making up those steps? Those are the nitrogen bases that are paired together. Then 2C is what parts of the nucleotide make up the backbone of the ladder. Well, the backbone is the outer edge of the DNA molecule. And when I look at that outer edge of the DNA molecule on both sides, I've got phosphate, sugar, phosphate, sugar, phosphate, sugar. So we have a sugar phosphate or a phosphate sugar backbone. Then question 2D, look at the bottom and top of the ladder. Are the rungs parallel? Do the ends of the strands match? So if I'm looking at the ends here, do they match? What's on this side? Is it also found on that side? No. I see there's a phosphate here, but there's not a phosphate there. And if I go down to the bottom of the DNA molecule, I would see the same thing here, except we're now on the bottom. There's a phosphate on the right-hand side and no phosphate on the left-hand side. And remember, that's because our DNA molecules are facing opposite directions. So on the left-hand side, it's kind of facing up, or that pentagon is facing up. And on the right-hand side, the pentagon is facing down. So since they do not match what's on the right side and the left side, our answer for question number question 2D is that these rungs, the bases, and the nucleotides are anti-parallel. They don't match each other. They run in opposite directions. They're parallel, so they run side by side, but the one side is pointing up and the other side is pointing down. So that's anti. Anti means opposite. And then question number 3 says that we need to label the bases on the model. So I'm going to come back to the model, and where those little question marks are, I'm going to replace those with the bases, and I'm just going to use the abbreviation. Adenine, I'm going to use A, thymine T, guanine G, cytosine C. So I find my first base is cytosine. I'm going to replace that question mark up here at the top with a C, and cytosine is paired with this light gray structure. That's a guanine. So I replace that question mark with a G. Then I move on to my next pair. I've got this striped shape. That striped shape is thymine, so that's T, and thymine is paired with the gray with the black outline with the little arrow. So that is adenine, so I'm going to abbreviate that A, and you continue that process the whole way down this chain so that you can see what bases are paired together through the whole chain. Then we are on to question number 4. When one nucleotide contains adenine, what base is adenine attached to on the opposite strand? So you're coming back to your model and you find adenine, or A, on your model. And if you're looking at, in my case, if you're looking at the right-hand side, you see that adenine is here on the right. What base pairs with it? And then you'll go back through and look at your other examples. And in every case, you should see that adenine pairs with thymine. So that's question number 4. Then we're moving on to slide 5 and question number 5. It says the 2 strands of DNA are held together with hydrogen bonds between the nitrogen bases. These are weak bonds between polar molecules. How many hydrogen bonds connect the two bases from question 4? Okay, well, question 4, remember this was looking at adenine and thymine. Okay, adenine and thymine. So I want to know how many hydrogen bonds are there between adenine and thymine. Okay, I'm going to come to my view. I'm going to make it a little bigger so we can see it a little better. And when I look at adenine and thymine, you should be able to see there are two solid lines between adenine and thymine. Those lines that are connecting those two shapes indicate the number of hydrogen bonds. Okay, so adenine pairs with thymine, and there are two hydrogen bonds that connect adenine to thymine. Hey, let me go back to my view so we can see the next question. And then we are looking at when one nucleotide contains cytosine, what base is attached to the cytosine on the opposite strand? So again, I'm coming back to my model. I'm finding cytosine, which is abbreviated with a C, finding what base pairs with it, and that's my answer to question 6. Okay, then question 7, how many hydrogen bonds connect those two bases? Okay, well, I'm looking again at my cytosine. Okay, I'm finding the base that it's paired with, and I'm looking for how many solid lines are there between those two bases. So you just count them up. Then we are moving on to question number 8. Use a complete sentence to write the rule for how the bases are arranged in the DNA ladder model. Okay, well, I'm going to start a complete sentence here, right? So in the ladder model of DNA, and I'm going to say a specific base, the adenine pairs with blank, and the guanine pairs with blank. Right? And you just need to fill in those bases to write a complete sentence. Okay, then we are moving on to slide number 6. Okay, it says, read this. Erwin Chargaff, an Austrian-American chemist, investigated the ratio of nucleotide bases found in the DNA from a variety of organisms. From his research, as well as the research of Rosalind Franklin, Maurice Wilkins, Watson, and Crick, developed the complementary base pair rule during their race to discover the structure of DNA. The complementary base pair rule states that adenine and thymine form pairs across the two strands, and guanine and cytosine form pairs. Okay, so if we use that information, we are going to fill out what are the complementary bases. Okay, if this is one side of my DNA molecule or one strand, A, T, C, C, A, G, what bases would pair? Okay, well, remember, adenine is abbreviated with an A, and this says adenine is paired with thymine. So adenine will pair with thymine. I'm going to put a T in that box that's directly across from it. Okay, then we have thymine. Thymine is going to be paired with adenine. Again, directly across, they pair together. That means there would be hydrogen bonds between these. So I'm looking at my DNA molecule kind of on the side. Okay, then I've got cytosine. Okay, cytosine is going to pair with guanine, right? Based on the base pairing rules that are described up here. Okay, and then you continue that process for the other bases here. Okay, question number 10. The ladder model of DNA is a simplified representation of the actual structure and shape of a DNA molecule. In reality, the strands of DNA form a double helix. Refer to the double helix diagram in Model 1. Describe its shape using a complete sentence. Okay, so I would start off. The double helix shape of the DNA is like a blank. Okay, so fill that in. What is it like? So I'm going to come back and look at the model. Okay, here's my ladder, and I'm comparing like the ladder to the helix. Well, what happened from the ladder to the helix is I took my ladder and I started twisting it. Okay, that's why you have these lines that are crisscrossing. Hey, they're crisscrossing because the ladder twists. Okay, and so it's actually a twisted ladder, or sometimes people describe it kind of like a spiral staircase. Okay, so those are two ways you could describe this double helix shape of your DNA model. Okay, and again, what makes up the steps of the model are going to be the bases, and then the black lines here in this helix picture, those represent the sugar phosphate backbone of the DNA model, okay, which are going to be on the outside edges of the DNA model. Okay, so then we are moving on to slide 7. In slide 7, you'll see that there is, this is a model of DNA replication. Okay, I've taken my DNA ladder and again, I've turned it on its side. Okay, and you'll see the bases are paired then in the middle here. Okay, and you'll look at the first pair, you'll see they're paired together. The second pair is paired together, and you'll see there is this blue circle around it. That blue circle is then labeled with DNA helicase. DNA helicase is a chemical that's going to essentially, like scissors, okay, separate the bases from each other. Okay, and this is what happens in DNA replication. So in DNA replication, your double-stranded DNA, okay, that's linked together by hydrogen bonds, DNA helicase is going to come along and is going to snip open that DNA molecule. Okay, so that's what this picture is showing. Okay, one strand is going towards the top, the other strand is going towards the bottom, and you'll see there are all these unpaired bases, okay, across your DNA strand. Those unpaired bases, the original strand that's here is going to be used as a template to then connect new bases. So if you look at the far right-hand side of the model, you'll see these are free DNA nucleotides. The complementary nucleotides, remember like thymine would pair with adenine, guanine would pair with cytosine. The complementary nucleotide is going to come along and pair up with these unpaired bases. So as the DNA opens up, new bases come in and pair, and we'll end up with 2 new double-stranded DNA molecules. Okay, and they'll be exact copies of each other. So essentially what ends up here at the top is exactly what ends up here at the bottom. Okay, so question number 8. Number the steps in order to describe the replication of DNA in a cell. Okay, well, the first thing that's going to happen, okay, if I look at my picture here, is this DNA helicase is going to come along and snip those hydrogen bonds. So that's step number 1. The hydrogen bonds between the nucleotides will break. Okay, step number 2, okay, once they break, okay, you're seeing that one strand is moving towards the top, the other strand is moving towards the bottom, so those 2 DNA strands are separating. Okay, so that's step number 2, the DNA strands separate. Step number 3, okay, free nucleotides are going to be attracted to those exposed bases. Okay, so this is an exposed base. The complementary nucleotide, complementary free nucleotide is going to be attracted. It's going to be pulled towards that. And like we see in the 2 bases that are on the far right, okay, the complementary bases will then pair with this unpaired nucleotide. Okay, so step number 3, free nucleotides are attracted. And then step number 4, okay, hydrogen bonds are going to form between those nucleotides. Hey, what's the rule used to join free nucleotides to the exposed DNA? It's the same rule as with all DNA base pairing. Okay, so complementary bases will pair together. So if I look here, okay, when I have an exposed base, if this is guanine, guanine will pair with a free nucleotide, and the one it has to pair with is complementary, right? It fits together with it. Okay, that complementary base, if this is guanine, it would be cytosine. Okay, if it's thymine, okay, like in this case, thymine pairs with adenine. Okay, so the rule is that complementary bases will pair together. Okay, and then question 14, this type of replication is called semi-conservative. Okay, think about the meaning of those two words. Semi means half, conserve means to keep. Okay, explain why DNA replication is called semi-conservative. It's called semi-conservative because half of the DNA is going to be old DNA, right? So we start off with this double-stranded DNA that gets pulled apart. When we make our new DNA, half of each new DNA molecule is going to be old DNA. And then the other half is the new DNA. Okay, so semi-conservative, half of the DNA is old, half of the DNA is new every time we replicate the DNA. All right, and then our final item on here. Once we take a look at our DNA replication, the last slide here, how does semi-conservative replication prevent mutations? It's going to prevent mutations because, again, we have a template, and that template then is going to determine the order of the bases, and those bases are going to be complementary to that template. That means that the chance of there being mutations is very low because those bases will not want to pair together if they're not complementary. Okay, and then the last part of this assignment is looking at the percentage of each type of base in some different organisms. Okay, I did the first one for you. You'll see adenine and thymine are always going to be equal in amounts. Guanine and cytosine will always be equal in amounts. Okay, so if we have adenine at 28%, guanine at 22%, cytosine at 22%, how much thymine will there be? Well, thymine and adenine always have to be equal. So that means if there's 28% adenine, there has to be 28% thymine. Okay, in salmon, if there is 29% adenine and 29% thymine, the remaining DNA has to be cytosine and guanine. Well, if there's 21% cytosine, that means there also has to be 21% guanine. Then we get to wheat. This is a challenging one, and you got to do like a little bit of math here. Adenine and thymine will have the same amount, right? So adenine has 27%. That means thymine will have 27%. So you're going to take 100 minus 27 and minus 27. That amount is how much will be split between guanine and cytosine. Half of that amount will be guanine, half of that amount will be cytosine. And then the last one, yeast. If there's 31% adenine, we know adenine and thymine have to be the same. Guanine and cytosine have to be the same. All right, ladies and gentlemen, I hope that helped you, and have a great day.